So first of all, I'd like to thank the organizers for giving me an opportunity to talk about our works. So I'm working in National Institute of Genetics in Mishima, Japan. So Mishima is small but quite nice city and quite close to Tokyo area. Okay, just 1 hour from Tokyo by Super Express train, Shinkansen. And from our campus, we can see very beautiful Mount Fuji. Okay, so please visit NIG to meet our nice colleagues and also to see the beautiful mountain. Okay. So in my talk, I'll discuss the local chromatin nucleosome behavior in living cells. Okay. Okay. So our main interest is how a long strand of genomic DNA organized in the cell, how genomic DNA behaves in living cells. This is sort of classical textbook view. DNA is wrapped around the core histone to form a nucleosome structure. And this is my nucleosome. And for a long time, the nucleosome had been assumed to be folded into this very regular 30-nanometer chromatin fiber, zigzag or solenoid or whatever, and further helical folding. However, past 15 years, quite many evidences, including our cryo study and X-ray scattering analysis, have demonstrated chromatin is more irregular and variable structure and should be very dynamic. Okay? And now, so more and more evidence to support this concept are available from many labs. So chromatin is irregular, variable, and dynamic. And at first, I want to emphasize the importance of local chromatin dynamics. Okay? So local chromatin dynamics is important because the local chromatin dynamics reflect the chromatin organization or environment in living cells, so providing structure information. And second, the local chromatin dynamics can facilitate the chromatin accessibility. I'll briefly explain. Okay. So these are multi-color computer simulation reconstructing chromatin environment. This modeling was done by Koichi Takahashi-san at RIKEN. And red balls are nucleosomes and green balls are model proteins diffusing in the environment. And please look at this side. So if the nucleosomes are immobile, fixed in the space, and green proteins are stuck and can't move further. So this is problem. However, so with fluctuating nucleosomes, fluctuation size is about 20 nanometer, and even at the same concentration of the nucleosome, green proteins can move almost freely. Like this. So local nucleosome fluctuation can facilitate the chromatin accessibility. So important. Then we focus on local chromatin dynamics. Okay? So to measure the local chromatin motion in living cells, we have developed this single nucleosome image. So I'll briefly explain how it works. And first, one of the cohistone H2B was tagged with hero tag and then stably expressed in the cells such as HeLa, HCT116, and RP1. And as you may know, a hero tag can be labeled with hero tag ligand, TMR, or other JF dyes. Okay? So we can label individual nucleosomes. So with the excess amount of this TMR labeling, H2B halotag TMR locate not only euchromatic regions, but also DAPI-dense heterochromatic region like this, suggesting H2B halotag incorporated into nucleosome genome-wide. So this is a very important point. And for single nucleosome imaging, so we treated the cells with very, very low concentration of TMR ligand to get sparse labeling. And using oblique illumination microscopy or hydro microscopy, which can illuminate very limited area in the cells with very low background. So we can see the individual nucleosomes as very clear dots. You see they are fluctuating, and this data was obtained by my PhD student, Shiori Ida. And these dots showed the single-step photobridging, so dots suddenly disappeared, suggesting each dot represents a single nucleosome labeled by H2B halotype TMR. And we determined the precise center of these dots by Gaussian fitting and track their motion to make trajectories like this. And here, we can trace only H2B halotype incorporated into nucleosome at this time frame. Okay? So we can see the free histones in this time frame. Okay? So we can focus on the nucleosome behavior. And based on this kind of trajectory data, we calculated the displacement of the nucleosome. As you see, the nucleosome can move 80 nanometer in 100 milliseconds. So this value is 8 times larger than this nucleosome size. So I would say that's quite a big movement in short time period. And we determined, we also calculated the mean square displacement, MSD, of nucleosome. And MSD showed how the molecule move in certain time. Okay? And please look at this black plot. They look subdiffusive, suggesting nucleosome somehow constrained. Okay? And when we fix the cell with formaldehyde, cross-linking chromatin, motion severely suppressed, like this red plot. Okay. And to get more information on local nucleosome motion, we took a log-log plot of our MSD data. Okay. So the plot linearly fit well up to 0.5 seconds here, and MSD exponent is 0.45. But beyond this time, so we couldn't fit well, suggesting motion mode changes over 0.5 seconds. So we focus on this very short time period. And next, as a collaboration with our physicist Soya Shinkai-san at Riken, we did the nucleosome motion angle analysis. So we determined this theta angle of the nucleosome trajectories. And in this trajectories, we determine the theta 1, 2, 3, and 4, and put all the angles in that plot. Okay? Like this. So please look at this plot. So nucleosome motion angle distribution somehow biased toward 180 degrees, this direction, as compared to Brownian motion of a controlled particle. So this means that nucleosomes often pulled back to their original direction in some constrained area. And to quantify this, so we introduced asymmetry coefficient, AC values. So just forward fraction is divided by backward fraction, and we took logarithm. So more pulling back force show more negative values. And if the motion is isotropic, like a Brownian motion, so AC values showed almost 0. Okay? So we can do some quantitative discussion for the constraint. Okay. Then we looked at the nucleosome motion during interphase cell cycle from G1, S, and G2. And as you may know, during interphase cell cycle, G1, S, and G2, genome DNA doubles and the nucleus become larger, okay? Like this. So the question is, do these changes affect the nucleosome behavior or not? Then we look at nucleosome motion in G1 and G2 cells. Okay. So can you see the difference? Of course, the nuclear size is different, but how about the nucleosome motion? If you can see the difference, you're right. Okay? So MSD plot showed G1, G2 cells do not show significant difference. Okay? So the MSD plot quite overlapped. And AC value also very similar. Their AC value also very similar, G1 and G2. So not only G1, G2, but also S phase also produce very similar MSD plot. So no significant difference. And these results suggest the local chromatin motion is not affected by nuclear growth or genome DNA doubling during interphase cell cycle from G1 and G2. Okay. So let me summarize the first part of my talk. So our single nucleosome imaging revealed the local chromatin motion is quite steady throughout the interphase cycle from G1S and G2. So we call this the steady state, sort of constant chromatin motion. Steady state chromatin motion is sort of average nucleosome motion of many nucleosomes. Okay? So this average motion is quite constant throughout the interphase cycle. Okay? Yeah. Yes. Later, in the second part of my talk, I'll discuss the nucleosome motion in mitotic chromosome. Okay? Okay. And then what the significance of this steady state constant chromatin motion? As I discussed at the beginning, so nucleosome motion is highly related to chromatin accessibility. So we think this constant chromatin motion throughout the interphase cell cycle is important to keep constant DNA accessibility. So that means that cells can do their routines, such as RNA transcription and DNA replication under similar situation throughout interphase cell cycle. So for more details, please look up our paper. And so far, so we focused on interphase cell cycle. And in the second part of my talk, I will discuss the local chromatin behavior in mitotic cells. So the main contributor to this work is Kazuhiro-san with the help of Sachiko Tamura-san in my lab. Okay. So we also sparsely label the nucleosomes in mitotic chromosome like this, and then we can check the movement in G2 and mitotic cells. Okay. And as you see, the nucleosome in mitotic cells looks less mobile, but still they are fluctuating like this. Okay. And the MSD plot showed the nucleosome motion is more constrained in mitotic cells like this. And the MSD exponent decrease and the AC value also decrease. So this is HeLa cell data, not only HeLa cells, but also HCT116, human RP1 cells, and Indian Munjak DM cell, a deer cell which have fewer and very large chromosomes. Okay? And all showed similar tendency, suggesting constrained nucleosome motion is sort of general features of mitotic cells.
Just a short question. So during metaphase nucleosome state, you still measure the dynamics through the nucleosomes?
So you mean through means?
The data measured, okay. The data measured in metaphases.
Yes.
For the dynamics, yes. Is it still based on labeling the nucleosomes?
Yes. Yes.
Okay, so nucleosomes stay on the chromatin, on the DNA during
yes, very
metaphase.
Yes.
Okay,
right.
Thank you.
Okay.
And maybe the questions can be left for the 25-minute onwards, given that the talks are quite short and constrained. Okay, thanks a lot.
Thank you. And the next question is how do the constraints emerge during mitosis? So to address this question, we synchronize this HCT116 cell in G2 by CDK1 inhibitor RO3306 treatment, and then release them. And G2 prophase, prometaphase, metaphase, and anaphase. And then nucleosome motion is more and more constrained from prophase to anaphase. Okay. And once the decondensation starts at the telophase, so constraint reduced like this. Okay. And this plot is MSC values at 0.5 seconds. As you see, the nucleosome motion is more and more constrained from prophase to anaphase. The question is what constrain chromatin during mitosis? Condensations? Yes. So condensations are key players for mitotic chromosome assemblies. So in vertebrates, there are two condensations, condensin 1 and condensin 2. And both locate around the chromosome center, like axis. And the current view said condensin 1 makes small loops, like these red balls. And the condensin 2 makes large loops at the bottom of condensin 1 loops. Then to see involvement of condensates in this constraining process, so we did rapid depletion of condensates by oxygen-inducible Degron system, which was developed by Masato Kanimaki-san at our institute. As you may know, okay. So oxygen-inducible Degron system is a great system, and AID tagged proteins rapidly degrade with oxygen addition in proteasome pathway. And here, SMC2, common subunit of condensin 1 and condensin 2 were tagged with MAID and also mCLOVER to monitor the depletion efficiency. And then we stably express in H2B halo tag in this cell line. And thanks to AID system, just 1 hour after oxygen addition, condensin disappears, and then chromosome become very abnormal shape, okay? But still chromatin condensed. So please keep this in mind. And then rapid depletion of condensin, so upregulated the nucleosome motion like interphase level. So MSD exponent and AC value also increase like this. So the chromatin is still condensed, but the nucleosome motion is somehow upregulated to the interphase level. Then during mitosis, more and more condensin capture the chromatin fiber, preserved to make loops and constrain chromatin fiber, chromatin motion. Okay. Okay, and then what else constrained nucleosome in mitotic chromosomes? So because without condensation, still chromatin condensed. Then we focused on the histone deacetylation because Schneider nicely demonstrated histone deacetylation is a major contributor to mitotic chromosome condensation. They use this HTAC inhibitor, TSA. So TSA treatment induced hyperacetylation of histone H3 and H4 tails. This weakened nucleosome interactions and then they condense chromosome. So please look at this one, and the TSA treatment slightly decondense chromosome like this. So without condensations, chromosome shape become abnormal, and but still chromatin condense like this. And the double treatment induced very drastic decondensation effect, and the chromosome almost dissolved like this. And we did a similar thing, and we synchronized the cells in G2 and treated with HDAC inhibitor and condensation depletion. And the TSA treatment induced hypersaturation of histone H3, H4 tails, and then slightly decondensed chromosomes like this. And then knockdown of condensations induced condensed but abnormal shape, like this. Then nucleosome motion increased, TSA treatment increased nucleosome motion like this, but double treatment induced really striking increase like this. So the nucleosome motion is really upregulated, even higher, much higher than interface level. Okay. So these results suggest deassociated histone globally constrained chromosomes via nucleosome-nucleosome interactions. So the nucleosome-nucleosome interaction is very important for global compaction. Okay. And then finally, so we wondered how differentially condensations and nucleosome-nucleosome interactions constrain chromatin. So nucleosome-nucleosome interaction act on the whole chromosome as a global constraint, but how about condensations? So work locally because the condensation locate around chromosome axis or globally. So to address this question, we look at the chromosomes in TSA treated cells. So in this situation, nucleosome-nucleosome interaction weakened, then condensing effect must be more prominent. Okay. Then we extracted nucleosome information from our imaging data, and nucleosome motion outside axis, and then compared with the nucleosome motion inside axis. Okay. And what we found is nucleosome motion outside chromosome axis is upregulated with the TSA treatment as compared to the nucleosome inside axis. Condensation enriched region is still didn't change. So these results suggest condensation act locally to constrain nucleosome. Okay, so let me summarize the second part of my talk. So during mitosis, the nucleosome motion become more and more constrained, and the condensation locally constrain nucleosome, presumably to make loops. And the condensation behave like a molecular cross-linkers, like this, and the condensation can be a motor, but cannot increase the local nucleosome motion, at least in our short time scale. And then finally, so the associated histone globally constrain nucleosome via nucleosome-nucleosome interactions. So our paper was published a couple of weeks ago, so for more details, please look up our papers. And in addition to condensation and nucleosome-nucleosome interactions, we recently demonstrated macromolecular crowding or depletion attraction also contribute to the mitotic chromosome condensation process. So if you are interested, please read our papers, very recently published in PNS. Okay, so at last, I'd like to thank all of my collaborators listed here. My PhD student, Shiori Ida-san, is a major contributor to the first part of my talk. So with the help of Soya Shinkai-san at RIKEN. And Kayo Hibino-san contributed to the second part of my talk. So with the help of Sachiko Tama-san and Masato Kanemaki-san and Masato Takagi-san kindly provided us their cell lines. Okay, and Ashwin-san attended this meeting, contributed to the statistical analysis of nucleosomes. So thank you so much for attention. I'll be happy to take any questions.
Thank you very much. We have 5 minutes for questions now.
Hi, it was nice talk, thanks. So when you are measuring these MSGs, the local environment is inside a condensate, right? Or I mean, what's the situation locally? I mean, it's a condensate or a free, like chromosome? I mean, bimolecular inside a condensate droplet or the chromosome?
Chromosome, you mean mitotic chromosomes or interface? Which part are you talking about?
Yeah, so throughout the, so when it's more and more constrained, it's actually getting trapped.
So are you talking about during mitosis?
Yeah, yeah.
Okay.
Yeah. So I was wondering that MSD doesn't really tell you how the neighbors get actually exchanged. It tells you about the overall motion of the chromosome. So is there any way that one might look into how the neighbors are actually getting, like, neighbors of a nucleosome are changing or not? I mean,
Neighboring.
neighbors, let's say if you choose one single nucleosome,
Yes.
and you want to see whether the neighbors surrounding a particular volume of that nucleosome is changing, like nucleosome getting exchanged or not. I mean, is there any way to tell that?
So we don't know the neighboring because of the diffraction limitation. We can only sparsely label
Right.
nucleosomes, and then we can just do some population analysis.
Right.
Okay, but your point is how the neighbor is behaving.
Yeah. So the idea is,
yeah, to answer your question, so we have to do dual color labeling. The two colors, so side by side, and then we can overcome the diffraction limitation.
yeah. So the point is the chromatin is clearly not frozen, right? I mean, it is having motion during the mitosis, right? I mean, from your data.
Yeah, constrained, more and more constrained around the chromosome center. And then nucleosome interaction also important to constrain chromosome level.
Okay, thanks.
Chromosome level. Thank you.
A lot of questions in front.
Okay.
Yeah.
Interface. Yes.
Focus on the first half a second dynamics, but you show that after the half a second, you start to change of MSD regime. And do you observe difference in the cell cycle phase after, I mean, in the later time regime? Because that's likely the regime where you start to feel more localized environments, the compartments, the TADs, whatever. So.
Okay. So that's an interesting point, and maybe the nuclear space can be affected by longer time scale, I guess. But we haven't looked at the longer time scale because nuclear movement or cellular movement also can affect the longer time scale.
Okay, last question, Madan over there.
Hi, I had a comment and a question, but I'll be very brief. The comment was on the shape of the trajectories that you showed, right? That you said it was elliptical shape. Yeah, I just wanted to make a comment that for a random walker, both self-avoiding and non-self-avoiding, if you look at the shape of the trajectories, that is, draw the convex hull, then that will always be an ellipsoid. Okay, it's a surprising fact, but it's true, and this was proved through a series of papers in the 1980s by people like David Nelson, Tom Lubinsky, etc., etc. Yeah, so that was just one comment that even the simple random walker unexpectedly gives rise to ellipsoidal in 3 dimensions, ellipsoidal shape. Yeah. The question that I had was, I might be mistaken about this. So please do. So many years back, in collaboration with Shiva Shankar, we had looked at the microreology of particles embedded in the nucleus. And we looked at in particular, we looked at the dynamics of histones and using rotational fret, time-resolved fret. And what we found there was that there were at least 2 populations of histones, those which are unbound and those which are bound to the chromatin. Now, when we looked at the mean square displacement,
only bound fraction, very stable.
You're only looking at the bound fraction. Okay, thank you. That was the question. Yeah, thanks.
Thank you.
Let's thank Professor Mashita again.